Yes, good evening to everyone. So, welcome to chapter 11 on T cell epitope prediction in internship and certificate course in artificial intelligence and machine learning approaches of drug and vaccine design. So, before stepping on to today's session on T cell epitope prediction, can we recap the last session contents? Yeah, what are the last session topics? Yes, very good. So I got from two participants, excellent. So we have discussed on immunoinformatics, like we have seen different types of informatics like bioinformatics, chemoinformatics, immunoinformatics. Bioinformatics is something related to biological molecules. If you work on, if you study the informatics on the biological molecules, okay, it is bioinformatics. If you study on chemical molecules, it is chemoinformatics. If you study on immune molecules, immune protein molecules, it is immunoinformatics. So, and we also discuss something on reverse vaccinology. The reverse vaccinology is a part of a study in immunoinformatics. And then we have discussed a reverse vaccinology. We majorly come across on design of peptide-based vaccines. Then we have discussed what is vaccine and what is adjuvant and what is the significance of both. Then we have discussed on the types of vaccine, and then we have discussed on T cell epitope, B cell epitope, and then we have also discussed on MHC molecules and role of MHC molecules. And we have seen MHC is generally a name of protein. Okay, and the corresponding genes of MHC, that is major histocompatibility complex, is HLA, that is human leukocyte antigen. If major histocompatibility, those who know the uh, immunology, no problem. But if they are very new to immunology, okay, major histocompatibility complex is the name of a protein, and human leukocyte antigen is the name of the gene. There's a like okay, so these genes code for this kind of a proteins, and these genes vary with person to person. Even siblings have distinguished human leukocyte antigen genes. Like how each and every person can vary with fingerprints. Okay, like similarly, even these genes signature varies from person to person. This is what we call it as gene phenotyping. Okay, and this is what we have discussed, and we have also discussed something on multi-tope vaccine. Okay, so very good. So whatever left out also taken by another participant. We have discussed on two examples in reverse vaccinology. That is, initially we have discussed that meningococcal polysaccharide vaccine is the first vaccine developed from reverse vaccinology. Then we have discussed on Nipah virus. I told you that I worked on Nipah virus and I also published in a journal. I spoke on Nipah virus protein W. Okay, and then I've gone across SARS-CoV-2 spike glycoprotein and was mentioned that there are four structural proteins and 16 non-structural proteins in the same SARS-CoV-2. And then we have discussed on the multi-tope vaccine called COVAX. Very good. We have discussed on COVAX, and I told you that COVAX vaccine has given nearly 99 percentage efficacy in patients when compared to Covishield and Covaxin. This is a good kind of a vaccine emerged, developed by US. Okay, I mentioned about that. And then we have discussed on types of epitopes like T cell epitope, B cell epitope. Okay, and even in the T cell epitope, we have seen there are two types of T cell epitopes and there are two types of B cell epitopes. This is what we have discussed, isn't it? And then we have seen a pathway present, how the MHC molecules are presenting and processing. Okay, there is actually like processing and presentation. Okay, an MHC molecule involves in a biological function called processing and presentation. So, how this is presented, we have seen an example with MHC1. Okay, and then we have seen more about the helper T cells and cytotoxic T cells. Okay, very good interaction. So this is what we have seen, and even I got this information from the third participant. So now I think in this session, three participants have given a good kind of an interaction. I anticipate that even many participants can come up with these kind of an interactions. Okay, so thank you all the three participants who have interacted with me. That you are showing the signs that you are following my presentation. Even I anticipate from the others also. Yeah. Now let me go to the T cell epitope prediction because I want to know whether it is reaching to your mind or not. Because many of them are even from other disciplines, but those who are in pharma, this is mandatory to understand. Okay. So one of the computational, you have to teach even the computational field professors. Okay. So something about this T cell epitope. First, we discuss on the workflow. T cell epitope database, MHC molecules. Are going to discuss on role mechanism, genes and function, difference between MHC molecules, immunological database. So this is what we'll be discussing it on today. Now let me just recap. I told you there are types of T cells. Okay, one is CD4 T cell, the other is a CD8 T cell. Okay, and CD4 T cell generally interacts with what type of molecule? MSC2 type of molecule. If it is an MSC2 type of molecule, what type of cells these are coming under cytotoxic T cells, isn't it? And these are helper T cells, CD8 interacting with MHC1. And then we have seen the structure of T cell receptor and B cell receptors. So these T cell receptor, B cell receptor structure varies with each other. And if you see in this T cell receptor, we have this as a variable domains. In this variable domains, the antigen is going to bind here. Okay. And if you see uh, if you see this kind of a T cell receptor, okay, we have four CD3 molecules. If you have a CD3, if we have a T cell receptor, we have four CD3 molecules. If you see in our top view, but if you see in the side view, we can see only two CD3 receptors. You got it? So actually, this T cell receptor is bundled with these kind of receptors, and generally in this side, you can able to see the antigen is going to bind here. Okay, so we can able to see this information like this. And now, if you see this CD4, okay, I wrongly mentioned the CD4 interacting with MSC2 is what the helper T cell. Okay, this is what we call it as a helper T cell, and this can be of two types. Once again, helper T1 cell and helper T2 cell. And related to cytotoxic T cell, it is CD8 T cell interacting with the MHC1 molecule. And if you see this helper T cell, just because this antigen is coming, okay, in and out, okay, so we can find the length of this amino acid epitopic peptide. Okay, this will be a large when compared to the cytotoxic T cell. The helper T cell is around 13 amino acid, and this cytotoxic T cell is around 9 amino acid. Okay, because the shape of MHC2, we can find two different subunits like alpha 1 and beta 1. So if and if the antigen is coming inside, okay, inside and coming out, then only it can perfectly bind with this CD4 T cell receptor. So here you find the 9 amino acid, and here in outermost you have 2 amino acid, and similarly here in this outermost you have 2 amino acid. So 9 plus 2 plus 2, so here it is 13 amino acid. But the alpha 1 and alpha 2 are the from the same subunits of MHC1 according to the structure of MHC, so you can have a smaller size when compared to MHC2. So this is how we can able to. So while designing the epitopic peptides, we fix this length for our design. And this I already told you in dendritic cells we have plenty of surface receptors around 162. And if you see DC T cell interaction, only these two are most important. One is MHC1 interacting with CD8 and MHC2 interacting with CD4. Okay, so this interaction is important along with the co-stimulatory molecules, that is CD80 or CD86 interacting with CD28. Any of these two molecules can interact with CD28. So if it interacts, the T cell gets activated. If it interacts with the CTLA4, instead of CT28, if it interacts with the CTLA4, it is going to inactivate the T cell. So either this co-stimulative molecules like CD or CD86 interact with CD28 or it interact with CTLA4. Okay, both the things won't happen at the same time. Okay, so this is something about T cell activation and what is the beneficial of T cell activation. If T cell is getting activated, the T cell is going to proliferate and it's going to produce two different T cells, isn't it? And we have seen this MHC1 pathway presentation. So just now I've told you, no? So like how the size of the peptide varies with the class 1 and class 2 because of a same unit alpha 1 and alpha 2. In the class 1 we have a small size, but here you have a larger size in class 2. And if you see this MHC genes, this is most important because today I'm going to take you the practical session. For this, you should understand this concept. That is in class 1 MHC, okay, this class 1 MHC, we have molecules like HLA-A, HLA-B, and HLA-C. There are totally three genes, okay? And these three genes encode glycoproteins expressed on the surface of all nucleated cells. That is the major function of the class 1 gene is to present the peptide antigens to cytotoxic T cells. Okay, cytotoxic T cells are actually CD8 T cells, okay, which interacts with MHC1. And class 2 MHC genes encode glycoproteins. If you see this class 2 MHC genes, this is generally of HLA, TP, HLA-TQ, okay, and this HLA TR. Okay, so these are the three genes we can able to find in human leukocyte antigen. Here, if you see, these also include glycoproteins, but these are primarily expressed in antigen presenting cells. Okay, and they present processed antigenic peptides. Here, it is for presentation. Here, we see the processing, and this happens in Th cells. It means that wherever you find the CD4 marker in T cells, okay, the CD4 marker is generally present in helper T cells. Okay, so this is the main difference between the MHC1 and MHC2. MHC3 is not that much significant, but these genes are expressed in the complement system. This is also a part of an immunity. This also contributes to the immunity, but these class 1 and class 2 MHC genes primarily contributes for the immunity. And if you see about the difference between the major histocompatibility complexes, okay, I told you we majorly have class 1 and class 2. I told you that the major difference is responsiveness with respect to T cells. The class 1 is related to CD8. If I tell CD8, it is CD8 positive. If it is CD8 positive, it means that it is CD4 negative. Here, if it is CD4 positive in class MHC2, it means that this is CD8 negative. So we have to understood like this. And co-stimulatory molecules, it binds with the CD8 co-receptor. Here it binds with the CD4 co-receptors. So to study, to know about this, okay, you can find in plenty of textbooks. There are so many parameters like sources of protein antigens, enzyme responsible, site of peptide loading, molecules involved in transport of peptides. So in all these parameters, okay, you can see distinguished features of class 1 and class 2. You can find the variations with the class 1 and class 2 in these parameters. So more about this, you can study from immunology books. Not only that, we can also see the MSc1 and MSc2 varies with respect to the structure, types of APCs, nature of antigen presenting, size of peptide. So everything it varies. And according to the textbook, the MHC class 1 is around 8 to 10 amino acids, but we can fix one particular length like a 9 amino acid. Okay, and similarly here they mentioned like 14 to 18, but we try with the 13. So whatever I've told you 9 and 13, this I just got the information from the John Hopkins. Okay, even if you enroll in any courses, even the John Hopkins hospitals are bringing this kind of a length of peptides. And now let me go to the next MLC class 1 and class 2 major types of genes. Okay, so if you see this HLA genes, we have ABC. Okay, that is the are coming under major genes. And if you see this minor genes, we have HLA, DP, DQ, and DR. Okay, so these are the class 2 MHC genes of and if you see the minor genes, okay, so we have HLA-E, HLA-E-F, and HLA-G, and the minor genes are HLA-DM and DO. And the HLA genes, if you see, we can able to find in human chromosome 6, and you can find ABC, or the genes you can find this, these genes are present in the specific locus of the chromosomes. So this is the short arm, and this is the long arm of the chromosome 6. You can find the DPQ D are located in this kind of a locus, and whereas ABC are found in this locus of this chromosome 6. And if you see the involvement of HLA with respect to diseases, HLA with respect to autoimmune diseases, this HLA allele, that is HLA-B27. I just mentioned you HLA-A, HLA-B, HLA-C. These are the major genes present in MHC1, isn't it? So other than that, if you see these in these genes, we have multiple alleles. Alleles you can find in the form of a numbers. So you can find a B27, even sometimes you can find B27 colon 01. You know what is that B 27 colon 0102? These are specific even up to the community. The B27 may be specific to a region, can be a country. And if you are specific even to the community of, okay, then we can also give like colon 01, colon 02. Like this, we have plenty of alleles. Okay. And if you majorly see this classification of HLA B27, they are more prone to ankylosing spondylitis. See, we know something like astrology. Okay, in India, we are following astrology, isn't it? We call it as Jatakam. Okay, so in Jatakam, we can find everything what is going to happen in future, what is happening in present. That's what many believe, isn't it? But the actual Jatakam, what Patrika, whatever we tell, the genome Patrika, okay, so that also can tell that even through these allele persons, so those who have these kind of a genes, they are more prone to get these kind of a diseases, ankylosing spondylitis, reactive arthritis, acute enteritis. Similarly, this HLA-P447 allele persons are more prone to get these kind of autoimmune diseases. And similarly, HLA-DR2, HLA-DR3, DR4. So you can find even the diabetes mellitus is happening just because of that gene. Not only that, rheumatoid arthritis most happen occurring disease like rheumatoid arthritis, diabetes is just because of these genes. Okay, so now we can realize the importance of the studying the HLA genes. Now let me take you to the server. I mean tools. One such the popular tool is a Raghava web server. In Raghava web server, we have tools like for a T helper epitopes. We have softwares like MHCB1, ProPred, okay, HLA-DR4, MHC, MHC2Pred, MHC Bench. So these are some of the softwares for T helper epitopes. So one has to go to this URL mentioned in bottom. You know, this is a server developed by India. A scientist called Raghava has developed so many tools for predicting T helper epitopes. Okay, also we call it as a MHC or HLA class 2 binders. And similarly, he also developed for the cytotoxic T lymphocytes. Okay, so we also have plenty of tools for that. So each and every tool can give a significance of the thing whatever mentioned here. So one has to go through this to first know the purpose of using the tools. In all these tools, the commonly if you have to give a input of a viral protein sequence. If you are finding, if you want to bring a kind of a vaccine for an virus, that is antiviral vaccines, then you have to feed the antigen of that specific virus. And if you want to bring the vaccines for cancer, we have to feed the cancer antigen. Okay, then we can able to predict here in these tools. Now, if you see this ProPred, okay, in this ProPred, so that is one of the tool from Bhagava web server. Okay, here you can able to predict the epitopes of MHC class 1, And for MSc class 2, we can able to go with ProPred. So ProPred 1 for MSc class 1 and just ProPred is for MSc class 2. So simply you have to paste the sequence here. Whatever you take, we take from the UniProt or NCBI. Okay, if you have to paste the sequence, the protein sequence of the virus or cancer antigen, whatever it is, whatever we are working, and then we have to simply select these molecules. These are the HLA allele molecules. So and then we have to simply submit it, and even we can fix the size of the peptide, whatever we want to predict it. And similarly, even for the MSC too, we can select the alleles, fix the peptide, and then we can submit the sequence. And like this, we can go with the net MSC pan. Okay, so for this, we can go to this website and just paste the cancer or viral antigen. We can predict it. And this is net MSC pan 4.1, the same thing. So these are some of the parameters. If you want to change the parameters, we can change it and we can predict the epitopes. So this is the major database. This is the yeah, everyone, all the researchers try to first explore on this kind of a database for discovery of peptide-based vaccines. Okay, this is actually Immune Epitope Database. IEDB stands for Immune Epitope Database. This is the largest database where if you see, we can able to see in this database presently 16 lakh peptide epitopes are deposited. We find so many T cell assays, B cell assays, or that is wet lab techniques, whatever we use for depositing the epitopes, we can able to see here. And if you see how to search for the kind of data here, so we can able to search even based on the host. Okay, if one is working on humans, we can search the human kind of data. Okay, like the data is related to pertaining to the disease. One can check with any kind of autoimmune disease. Like you can type for asthma, you can type for arthritis. Like if you select the human and if you select the arthritis, okay, uh, then obviously whatever alleles responsible for human arthritis, we can able to find it. And not only that, other than alleles, we can also find the peptides. Okay, so what are the peptides we can use for the treatment? So based on alleles, even we can screen the peptides and we can choose the peptides. Okay, and generally the EDB immune epitope database. I told you this is used for designing of peptide-based vaccines, isn't it? So not just peptide-based vaccines, even precision-based vaccine design can be done using this IEDB. Like if I can directly take you to the website and explain you. Later I can come to this PPT. It is got it. See, first I'm going to take you to this IEDB website. So this is actually Raghava web server. If you go to this crdd dot thing, we can able to see, we can get inside this. Okay, now I take you to this website IEDB. Okay, participants have any, if participants have doubts, you can ask me. Do you have any doubts up to whatever I have covered? Yes or no? Do you have any doubts? No, sir. Thank you. Because presently for this course, we have diverse kind of participants have participated from petroleum, chemical, biotech, pharmacy, MBBS, MD, BDS. So many types of participants have enrolled, even in the past course and even in this course. Definitely, it will be helpful. Even I know my friend who working in Ayurveda, who is working in naturopathy. Even these doctors have enrolled for this course. Okay, you can understand something, but if you want to make more understanding, then even the participants, those who understand, have to interact with me so that even the others can understand. Okay, so you are letting others to understand something, whatever they can make it. Okay, let me take you to the IEDB, Immune Epitope Database. Okay, Immune Epitope Database. So to get inside that, we have to go to this URL, iedb.org. Okay, so inside that we have a T cell epitope prediction here. If I want to go for a T cell epitope, we can either go with MSc class 1 or you can go with MSc class 2. Even in MSc class 1, we have immunogenicity, processing, binding illusion, and a class 2, we have similarly like this. Okay. And if I want to work on these kind of tools, not only this is related to T cell epitope, before going to T cell epitope, I also work on this population coverage. These are additional tools one can able to find in epitope analysis tools, population coverage, conservation, epitope cluster tool. Initially, I just work on this and then I go with this data. Okay. And even before working on this, first we have to take a sequence of a cancer antigen or a sequence of a viral antigen, which is the input for these tools. So if you want to take an input, then obviously we have to take from initially we have to take from NCBA, okay, or we can take from Uniprot. If you see my PPT, okay, somewhere I have mentioned this sequence. If sequence is not there, we can take. Okay, there is no sequence, so better I can take from the Uniprot. So Uniprot, I am going to take a cancer antigen. So if Nipah virus is there, isn't it? So first I type like Nipah, or if I type Nipah virus, it directly take you numerous proteins of Nipah virus. No, yeah, I'm getting some Nipah virus. If I want to get a specific, like if I want to go with this RNA directed RNA polymerase, the gene is L, but I worked on W. But if you want to work on L, this is actually very large, 2244. It takes more time. So better I take a small kind of a protein. The small kind of a protein I'm going to take is a G, glycoprotein. Of course, nucleoprotein, the size is very less, but the glycoprotein is a significant target, I believe. So I'm going to take this. So I'm going to take a Nipah virus glycoprotein. Okay. So this is my target for for T cell epitope prediction as well as B cell epitope prediction. What I'm going to do it on tomorrow. Now for taking a sequence, click this sequence. So after clicking this sequence in this UniProt, click download. Now we get the entire sequence. Either paste the sequence or even take the entire pasta format of the Nipah virus glycoprotein. After that, okay, so if you want to feed it in T cell epitope prediction, you have to click here and you have to paste it. Okay, so now I just click here in MLC1. Okay, so if I click this MLC1, I got a default is SARS glycoprotein. If you want to try, you can try with it, or if you want to try with our kind of a molecule, uh, I'm going to paste this. So I am going to replace SARS-CoV-2 glycoprotein with a Nipah virus glycoprotein. So peptide length for default I kept is 9. I'm not going to change anything. But if you want to fix the alleles, if you want to fix what alleles we have to feed, one can able to go with population coverage. There is one tool called population coverage in the epitope analysis tools. Just click here. Now I am directly taking you to the population coverage tool. So in this population coverage tool, you know what I'm going to take. So if I want to take a specific community people, for example, Tamil Nadu, if I want to design a kind of a peptide based vaccine for Tamil Nadu Nadar community people, whatever I'm going to design, it is specific. It is specific that it can able to cure for sure. Like how I told Covax can able to cure 99, can give a 99% efficacy like that. Okay. But if you see whatever vaccine we designed for Tamil Nadu Nadar community, it won't work for the USA people because allele definitely it varies with the region also. Okay. So first I'm going to check with designing a peptide based vaccine. For this, I'm going to click now. The next step is to click the allele frequency net database. So in the population coverage, I have a URL called allele frequency net database. I click that. Now we got this kind of an URL. So after getting inside this allele frequency net database, we have to click populations. So if you click populations, okay, we have probes populations by geographical regions. Click this. If you click this browse population by geography, this is stepwise procedure to select the community people and to know what alleles, what are the dominant alleles of the specific community people. You can take anything. You can take Telugu Chettiar, or even we can check the select the Bengal. Okay, whatever community we want to work, we can work on that. You know, if you see this region wise, we have see major continental regions, isn't it? So if you want to check the India, India, I believe it's located in South Asia. So in South Asia, HLA, nearly 69 population is there, isn't it? I'm just going to click this 69. Ah, yes. If you go to the South Asia, we can see the India. If you go somewhere, we don't find. Okay, fortunately, we are in South Asian. We can able to find all the Indian people. If we are working on a vaccine design for an Andhra Pradesh Brahmin, so see, this is the dominant allele for them, TRP1. Okay, if I am working on this Tamil Nadu kind of, so Tamil Nadu, Chennai, Dravidian, if I want to work the Chennai people, okay, if I want to design for a Chennai, then I can go with this Tamil Nadu Chennai. So the dominant allele is DRP1, but even in DRP1, we have frequencies, so we call that is allele frequencies. Go inside. See, stepwise, I'm guiding. So almost we have gone up to 7 to 8 hyperlinks inside. Now we can see these are the Chennai people, and the Chennai people alleles or this. So we can see allele like from 01, 03. If you don't find TRP1, 02, we have 03 after 01. You know what is this DRP1? I told you DR, DP, DQ, and DR stands for MHC2 alleles. So these are MHC2 alleles and specific alleles 013, 04, 07. Okay. So like this, I can able to see if you see percentage of individuals having the allele, the most dominant allele in Tamil Nadu, Chennai is DRP107. 30.6% individuals have this. So if you are designing any kind of peptide based vaccine, can be for Nipah virus, can be for SARS-CoV-2, focus on this kind of an allele. Okay, and then we can also check for your kind of region and the community also. Okay, and the next to that is this thing. So now I am going to choose DRB107, and the next is DRB113. Okay, you don't find any MSC1 alleles, but only MSC2 alleles are dominating. So now I'm going to try with this, but see, this is MSC class 1. We can't able to select that kind of an allele. We can select only HLA, PC. Only if you try with the class 2, then only we can select that. So we don't have any kind of a class 1 molecule here. Got it? So if only if you go with this class 2, so the class 2 is here, okay, then you can get this kind of a window. Now I'm going to paste this Nipah virus glycoprotein, that is the input, and defaultly it is in 15. Either you can fix it as 15, no problem, but in your entire research you have to fix 15, or you can reduce to 13. Okay, now if I keep like this, you have 13 to 15 size, just because I've dragged. So you can also have a range kind of a peptides. So 13 to 15 amino acid epitopic peptide, you're going to search now. And now here, if I want to search only for the Tamil Nadu Chennai people, okay, what I have to give? So whatever I found out, HLA DRB 107, isn't it? So type that, or even you can use allele finder. So here HLA DRB 1, okay, in DRB 1 asterisk 07. Yes, we have 07. So 07 we have 1, 3, 4. Numerous things are there. But if you see in this allele, 07, only we have 07 alone. But in 07 itself, we have plenty of alleles from 1 to 12. If you focus on the 7, we have to give everything. Okay, again, we have to type the same. HLA, TRP 1, asterisk 07, okay, colon, we have 0304. There are plenty of alleles to try. I just going to select the two, the top two. These two alleles, okay, the most dominant alleles. Okay, that alone I have selected now. Now click submit. So see here, first I have selected these two alleles based on our desired, uh, based on what community of population we like to target. Okay, so that alone I have selected, and I have fixed the range of 13 to 15 amino acid epitopic peptide. And now I am going to predict based on this kind of a default method, net MSC2 pan. And if you want to add any other prediction, you can make it, or if you don't, you want to just start doing this. Okay, I am going to click this run. We have a run button here. I just click run. So now we are going to search, okay, the MSC2 peptide, epitopic peptides, okay, for a size, for a length 13 to 15, for a, specifically for Chennai population. So similarly, if you want to try with the MHC1, now whatever we are trying is for MHC2. Okay, if you want to try with MHC1, but for Chennai population, we don't have dominant MHC2, so we can try with some other community. So I'm going to choose some other community in this allele frequency database. So and we try with that. Instead of Chennai, I'm going to select this time Andhra. Can we select Andhra Brahmin? No, still DRP1. Only Golla. We find A and B. So here itself we can see the HLA locale. Okay, where we can get this MSC1 alleles. So Kerala, Karnataka, North. Okay, so like this we have plenty. So if you see this New Delhi, we have ABC. Can we try New Delhi? Like however we have touched the Chennai, now I'm going to touch the New Delhi population. So allele frequencies. If you see this dominant allele in the New Delhi, according to the percentage of individuals, it's not displayed here still. So based on the scores, we have to decide. It's loading. So this is the dominant allele, A1101. So based on allele frequency, 0.2, so A1101. So now I'm going to paste the same sequence in MHC1. This is MHC1. Okay. And I'm going to take this allele frequency as A1101. So type that HLA A, what is that? 01, isn't it? 1101, sorry. HLA A asterisk 1101. Okay, I have selected the allele. Similarly, if you want to select the second allele, select the second allele and submit. Now I just selected only one allele here. Only one allele I have selected, and default size only one size. 9 amino acid peptide I'm going to search. If there is no other prediction, then simply you have this run button, click run here. So here also it's running now. Did we get any results in MSC2? Yes, we got the results in MSC2. So see the results in the bottom. Okay, this is how we can see the results for MHC2. So to download these results, we can download this. Download all rows. Either you can download in the form of tabulated separated value file, or even we can download as comma separated value file. These two file extensions we can open in Excel. Okay, so download this or that. Can I download TS with now at present. So I'm going to download this in desktop bioinformatics demo AML. So this is for MSc2. This is an MSc2, MSc2 kind of a TSV. So now whatever I downloaded that MSc2, okay, .tsv, I have to open it in Excel. I don't find Excel, so then I have to open Excel and I have to open Excel. Excel, I'm opening the Excel software. Now I am going to open that in a particular location. The location is in a desktop on AML. AML. All Excel files, all files. What is that? This .tsv. Oh, okay. Next, finish. This is the data. This is the output data, wrap text. So see here, we find, I zoom in and show you. So these are the headings actually. Peptide start and peptide length. Just because we have given length 13 to 15 amino acid, so you can find one in this range. And you know what is the most preferred peptide? What is the best peptide? The best peptide is shown at the top. And even in the score for that, you can see it is minimum. Okay. So, and this net MSC score is minimum for the best peptide, and it progressively increases. So, of course, if you see the base peptide, or even we can see, mm, I can open in the database. The base peptides can be visualized based on the Excel itself. I can able to screen it. So this is a net MSC2 score. Now I'm going to use data filter, and in this filter can we sort largest to smallest. So this is the largest actually. If this is largest, this is the best peptide. Okay, so this peptide is the peptide is EGF. So this sequence, okay, of MHC2 is the best epitopic peptide, okay, for the Chennai population. That is, if we want to target Nipah virus glycoprotein, we have to use this as a ligand, as a epitopic peptide. Okay, and similarly, if you go with this MHC1, this is what we tried. This is the result for MHC1. This I tried with the Delhi, New Delhi population. Okay, what is we just search for the best peptide for the Delhi population. For that, again we have to download this file. This time I'm going to download all rows in CSV format. So in CSV format, whatever I'm going to download, okay, this is nothing but MHC1 peptide in CSV format. I just saved it. After saving it, we have to open through Excel. Okay, so I'm going to open file, open. I'm going to use the browse, and I'm going to a desired location. The file name is MSC1 peptide. So just open it. So you can able to find if it is CSV, you can directly open it. Wrap text. I'm going to bold it. I'm going to zoom it. So you can see this HLA0, the standard, we can find only 9 amino acid peptide lengths. So many peptides are generated. If you want to see that total data, how much peptides are generated, I'm just dragging and showing it to you. So we have around 595 peptides are generated. Out of these 595 peptides, to check the best one, we can able to filter through net MHC. So actually, this is sorted based on highest to the lowest. So no need to sort anything. already it is sorted. So the best peptide for the Delhi population is this sequence AVY. So this peptide is used to target that Nipah virus glycoprotein. Got it? So this is for MSC1. The previously whatever is shown is for the MSC2. So this is how we have to search for the results using IEDB. Okay, in IEDB, if you recall, I just taken you to the how to predict MSC1 and MSC2 using the uniprot sequence, the Nipah virus W protein. Okay. Now let me take you further to the PPT. So we have discussed like a T cell epitope prediction. We should also need of these kind of tools like a population coverage. And if you see the MHC one, just a recap. So just paste the sequence of the viral or a cancer target in this location, and after that fix the length, and you click this run here. And similarly for MSC2, go to this tool, and then you can able to predict it, fix the alleles and predict it. Okay, and if you want to check the population coverage to know about the regions, for example, if you're targeting the US people, or if you're working in China and if you're targeting the Chinese people, then we have to even select that kind of a regions. So for India, it is in South Asia. How about for Japan? You have to select the particular region. Okay, and then only can find where the Japan is there, and then this is the thing I have initially taken you to check the allele frequencies. Okay, in the population coverage, I get inside and I got this information. And if you want to target both MHC1 and MHC2, even we can search for it. Okay, even we can search like MHC1 and MHC2. Simply have to paste the epitope sequence, like whatever epitope we have predicted now in MHC1, type the sequence of the epitope. And if you have that allele information, type that allele. That is, for example, HLA-A, for that you got that 9 amino acid peptide, isn't it? The New Delhi population epitope, that 9 amino acid peptide epitope, which is present at the top in the Excel sheet. You paste the sequence and you type the allele. Okay, like this, you can feed as many number of epitopes and feed that kind of an alleles. And if you can click submit, you can also get uh, the frequency, or like how effective or not. So this is another kind of a result you can try with the population coverage. And this is a publication already I've taken on yesterday, that I've gone with the T cell epitope based, isn't it? So for this, you can, I've also, if I didn't paste it in the WhatsApp group, I paste this kind of a journal publication. So see how I worked. See, even in this publication, I didn't tried perfectly. So for MSC1, we should restrict up to 9 amino acid. And for MSC2, it can be 13 to 15 amino acid. But you know what I did? I did a small mistake, that even for MSC2, I tried with the 9 amino acid peptide. That should not be done actually. For MSC2, that should be minimum 13 to 15, or even it can take even up to 17 amino acid. Somehow I tried, and even I got published. But if you are trying in future, you're correct. Like you should have a more length for MHC2. And more about this kind of enzymes and the enzyme inhibition for the types of viruses, you can check from this book chapter. Okay. And workflow I already mentioned in my study. I also mentioned for a SARS-CoV-2, but for a Nipah virus, for the we have structural proteins like N, P, M, F, G, L. So all these structural proteins, so they have already tried in 2018 itself, in 2018 to 19. So I have only one chance to try, that I can able to try only with the W. So that's what I tried. Even C and W, they have already tried. I mean, tried, it means that they have already tried and they have already published in repeated publications. Okay, so if you have any chance to work on any kind of a new viruses where you don't have any pre-published data, so you can try with this reverse vaccinology. Okay, this is something about how to bring a kind of a precision-based vaccine design. Like now, I just demonstrated for Chennai population and New Delhi population how to uh, how to design peptides for a specific virus. We can even try for cancers. Okay, with the cancer antigens. And in T cell epitope prediction, it's not just limited with the MSC1 and MSC2. We can also predict something called as TAP, okay, transporter associated with antigen processing. We can also check features like amphipathicity, motifs, etc. So for this, they're using the machine learning technique like ANN support vector machines. So one such is T cell immunogenicity. For checking T cell immunogenicity, one has to go to this tool. So in T cell immunogenicity, the immunogenicity is predicted through antigen processing, peptide binding to MSC molecules, and recognition by a cognate T cell receptor. Okay, so immunogenicity is calculated like this, and conservancy is a fraction of the protein sections that the epitope contains, and identity is a similarity. So that is, if you want to check how many epitopes, the epitopes are common to a particular kind of a protein, we can able to check through the conservancies. Okay, and toxicity. So after predicting the epitopes, one has to predict whether the epitope is toxic or not. If it is toxic, even though it shows as a best hit for MSC1 and MSC2, we should ignore. So we have to go with the second and third epitopes. We have to check what epitope is showing a non-toxic. One is a non-toxic epitopes alone. We have to screen and we have to show that as a best epitope. Okay, for predicting the toxicity of epitopes, the physicochemical properties of peptides like hydrophobicity, hydropathicness, that is hydrophilicity, molecular weight, net hydrogen, side bulk, and steric entrance are taken for calculation of toxicity. So in toxicity analysis, one can check only toxic or non-toxic. You won't get any values here. If it is non-toxic, we can consider. If it is toxic, ignore. So for this, again, for predicting the toxicity, we can go with the Raghava web server. So how to go with the working on a sample? In my publication, I worked with Nipah virus W protein. I mentioned, isn't it? So even in the publication, you can see the sequence. So if you have the publication, just copy that sequence and paste it. But you know what strategy I used? I didn't try with a single server. I tried with multiple servers. So these are something related to IEDB. These servers are present in IEDB itself. Okay, that is, I tried with MSC1 and MSC2. Okay, in IEDB. Other than that, I also tried with the Raghava web servers like ProPred. IEDB is an exclusive web server, and even NetMSC MSC2 is also there in IEDB, and we also have an exclusive web sources also. So if we have plenty of web resources for checking MHC1 and MHC2 peptides. So in my research, I just focused on, other than IADP, I focused on ProPred tool. Okay, and I just got some kind of an epitopic peptides. I just fixed 1, 2, 3, 4, 5, 6, 7, 8, 9. For all the MHC1 and MHC2, I fixed as 9 amino acid. Okay, I fixed as 9 amino acid, and but it should not be done as I mentioned. Okay, we should have a 9 or 13 amino acid, at least 13 amino acid in length. Then only we can able to screen. I just got like after comparison of multiple servers, so which server shows a similar kind of a result. So that I have screened. I've screened this SP, this epitope, and this epitope. One epitope for MSC1, the another epitope for MSC2. Okay, like based on immunogenicity, based on conservancy, and based on toxicity, we have screened epitopes. Okay, so one for MSC1, the another for the MSC2. Okay, but when you do it separately, don't mix it. Okay, when you go for a publication, do separately for MSC1, do separately for MSC2. and do even for a specific community, or you can try with for a entire nation. For India you can try with multiple alleles, and then we can come up with a kind of a best peptide. Only thing is that we have to relaxly work. If you get thousands of peptides, we have to screen based on multiple parameters. Take these parameters for screening, or even take multiple servers. If you get top 20 for MSC for IEDB, you can get another top 20 for ProPred. So top 20 for ProPred and top 20 for IEDB, and then you can get which is the best from the which is common in ProPred and IEDB. If you get a common thing, and based on the score, you can filter which is the best one. If you have multiple servers, once again I'm telling, if you are using multiple servers for comparison, yeah, you can don't restrict up to the best single peptide in ProPred and IEDB, but you take top 20 or even top 50, and you check what whether these 50 peptides predicted from ProPred is matching with IEDB. If you get nearly some 5 peptides are matching, you check which is having a good score. So based on the score, you just screen the best peptide from these multiple servers. Got it? This is how we can compare with the multiple servers or even with the multiple parameters. And after screening the base peptide, okay, so this peptide, whatever I screened, this is for MHC2. This is for MHC2. So what I did is, and whatever I screened, this next epitopic peptide is for MHC1. Okay, you do separately, then you won't confuse. So if I get this as a base peptide, okay, so I have to check whether this whatever peptide I designed from the Nipah virus W is talking well with the human HL allele or not. Okay, because whatever peptide we predicted for the MSC2, so this protein, this protein I have taken from NCBI, GenPept. So whatever sequence I have taken, I have gone for homology modeling using this modeler. I have predicted a protein structure. Whatever structure I have predicted, I try to dock my peptide with the protein. This docking can be done even with online servers. Okay, and if you do this kind of a docking, we can get this kind of a score. Okay, whatever score you can get, you are getting, you can display that. And similarly, so for designing the peptide, one can try with the PepFold 3 server. For designing peptide, we can use PepFold 3 server, or even we can use what other server I have mentioned you, Vika or something I mentioned you, isn't it, in my previous session. Use that. Even Discovery Studio also we can use for designing peptides. Design the peptide and get a 3D structure in the form of .pdb. Dock this peptide.pdb with this protein.pdb. Then you can get the scores. You can show the scores. Okay, so this is how one can able to check after predicting the peptides, after screening the peptides. Later we can go with the MD simulation. So whatever I peptides I just I've screened, and whatever I talked with this MHC2 molecule, the specific allele protein. Okay, so we can able to see the RMST here. The root mean square in an average we got around 0.75, and you can see in RMSF it is not touching 1, so it is stable. Whatever peptide docking with MSC2 is stable. Okay, and even we get area is a consistent area for certain time. Okay, this is how we can tell up to this second the area is consistent, and radius of gyration. So there is no much variation for this particular time, but after particular time we find the variation, and after 150 nanoseconds we can see again a stable kind of radius of gyration. So stability you can find in this time and in this time when peptide is binding with the protein. So this is how we have concluded after talking a peptide with the human MHC protein, allele protein. So this is how we conclude. And this T cell based vaccines, if you want to develop, other than designing the peptide based vaccines, if you want to develop the adjuvants, even we can able to go with this kind of an adjuvant based research. In my lab, we work on dendritic cells. The dendritic cells itself acts like an adjuvant. And in our lab, we are also working on plant-based adjuvants like AH03. We are also working on IL-2, IL-4, and GM-CSF. So we are developing these kind of an adjuvants for various kind of vaccines. So this is what the recent research happening in this decade. Okay, so previously, if you see the adjuvant, okay, aluminum salts, especially alum, aluminum sulfate was used as a most of the for the most of the vaccines. This is used as an adjuvant. But this is an inorganic kind of a compound. It has its own disadvantages. So nowadays, we are working on biomolecules like protein molecules or plant-based molecules or even cells can also act like an adjuvant, so which can increase the efficacy of our vaccine antigens. And this is one such database developed in my lab. So the PG students have developed a kind of a immunoanalytical database. This is for exploring on adjuvants for type of diseases. For asthma, which is the best plant-based adjuvant, and for rheumatoid arthritis, which is the best plant-based adjuvant. So like this, we have developed a database for the types of diseases. Okay, so and if you see, this is a kind of a research I've worked in my laboratory, in my PhD research. I've worked on two different plant-based compounds, and these plant-based compounds can work as an adjuvant, and whether these adjuvants can give a kind of a potential beneficial effect on T cell activation and proliferation or not, and whether the life cycle of dendritic cell is changed or not. All these things we have tried. Okay, so the ultimate result is the plant-based compounds or worked as a best kind of an adjuvants for developing T cell based vaccines. So adjuvants in COVID-19 vaccines, we have plenty of adjuvants like a spike like for a spike like a protein, polyethylene glycol is used, and even adenovirus can be used for this kind of an. And if you see for Sputnik, okay, the adenoviral vector is used as an adjuvant. So many examples are provided here for the vaccines. Okay, so instead of simply restricting up to designing peptide-based vaccines, one can also design the adjuvants for the vaccines. So I've completed today's session on T cell epitope-based prediction. Okay, so on tomorrow we'll see the B cell epitope-based prediction. Any doubts you can ask me at present. Do you have any doubts you can ask me? If no doubts, thank you very much for the interaction and the patience maintained in the case lecture.